ex vivo, we saw that the T cells responded to the metabolites. We went on and tried to show in vivo that the T cell metabolism was altered, or that the T were changed in response to exercise. And this was by far the most difficult experiment of the study. So what we did was that we had donors of T cells. That was mice whose T cells recognize a specific antigen. So in this case, the OVA antigen. So it's the OT1 model. So we took the CD8 positive T cells and we injected them into recipient mice. And 24 hours later, we vaccinated the recipient mice with dendritic cells expressing the OVA or showing the OVA, so that the T cells got activated. And then we waited for 48 hours for the T cells to have the time to start activation. And then we did the exercise bite. So we let the mice do a warm-up, and then we introduced the 13C labeled glucose via IP bolus. And the idea of the experiment was to give the labeled glucose to see if not glucose itself so much as metabolites of glucose that would come out of the muscle when the glucose is used by the muscle during exercise. And like, for example, lactate that we know of, but also some of the TCA metabolites. So that was our attempt to see if we can identify those metabolites in the T cells of the exercising mice. So after the warm-up, we introduced the labeled glucose. We allowed the mice to do an all-out sprint exercise. So we increased the speed of the treadmill until exhaustion.